Hello everyone, my name is Mina. I'm a PhD student in Institute for Computational Genomics at Rebetal University Hospital Aachen. I'm going to talk about multi-scale approaches for patient-level analysis of single-cell and spatial transcriptomics data. So many diseases have phase transitions. For example, myocardial infarctions going through different stages from healthy to ischemic stage. So through these stages, there would be some inflation, some dead cells, stress cells that would appear through all of these stages. So it would be important to identify these critical events that affect the prognosis of the disease and see what causes the transition from the chronic to acute states. And this would gives us to understand the molecular mechanisms for have a better predictions and have a personalized treatments. So deep molecular profiling techniques are multi-scales that the data measured for one patient is represented by distributions of the cells. And each cells is a distinct molecular state and has a particular cell types. So in this sense, how can we measure the similarities of multi-scale molecular profiles of patients? So by having this single nucleus RNA-seq and all the samples and getting the cell distributions of each patient, can we find the patient's specific clusters or trajectories? So I'm going to introduce here the pilot patient-level distances from single-cell genomics with optimal transport. So as an input, we get the clusters and the integrated single-cell RNA-seq, and then based on that, we get the distributions of this clusters of each patient separately, and having the similarities of the clusters using the optimal transport. We can find a transport plan for moving masses of cluster distributions of one patient to another. Then we can use a Wasserstein and distance to estimate the total cost associated with the optimal transport plan. The graph structure learning on diffusion map provides a disease progression rank. And so this disease trajectory represents the similarities of multi-scale molecular profiles of the patients. We can also, based on the distance of the patients, cluster them. So, but here I'm just focused on seeing them as a continuous one. So having this trajectory, can we extract the nonlinear changes at different levels of the multi-scale data that goes through this disease progression rank? Uh, then by understand this, we can have the features that cause the transitions from the chronic stage to acute stage. So here at the sample level, we were, for example, interested in the clinical parameters like age, different tissue structures, if there is any connection with the disease trajectories. At the cellular level, we check the cell types frequency, if there is a linear or quadratic and nonlinear changes with the disease trajectory. And at the molecular level, we find the expression patterns for each cell types, and we can further cluster them based on the patterns. Further, we can have the condition model to compare the expression between cell types. So here we can explain all of these changes by a model called a Hubble regression models, a nonlinear model. And for the condition model, I also use the Wald statistical test that I will explain in the next slides. So with the assumption that our features like clinical parameters, cell proportions, and gene expressions that have a linear quadratic or linear quadratic changes, um, changes through this disease trajectory, I will use a robust regression model called Hubble regressor, which lessen the effect of the outliers and find the fitted function for each features. And then based on some condition like F statistic and adjusted R square, we find the best function that explain the changes through the disease trajectory. For the conditional part, we interested to find the genes that are cell type specific when compare the expression to the disease trajectory. So I use a Wald statistical test with null hypothesis that the fit of functions of the gene expression between two cell types would be the same. So as you can see here, in each time point, the expression would be compared between cell types and will be measured the difference all over these disease progressions. So the condition would be through between two cell types or one cell types compared to other cell types. So as I talk about the myocardial infarctions, having these disease trajectories in the diffusion map uh, space, we can have a closer look at cluster changes in this trajectory. So we wanted to find cell type importance for disease, and we observed that the frequency of healthy cardiomyocyte linearly and stress cardiomyocyte quadratically decrease with the disease progressions. And macrophages and myofibroblasts had quadratically increased. So that was some hypothesis that shows that the cell frequency somehow related to the disease. So we can have a closer look at the myofibroblasts and uh, analyze the genes. And so I cluster the genes based on the patterns and find the four patterns of the genes. So we can say that there was a late expression, early expression, and in the cluster 4 there was a linear early expressions. So the other interesting things that would be the cell type specific genes. So here's for the cardiomyocyte, we find that the genes that higher express in cardiomyocyte compared to other cell types. So we realize that HDAC9 is a repressor of the cardiac hypertrophy, and MLIP is a regulator of cardiac functions. And for the myrofibroblasts, we find two genes that are specific to these cell types called COL1A2 that shows the increase in the matrix production, and DCN is shows the loss of expressions and it's an anti-fibrotic gene. So at the end, I should say that the pilot is an interpretable framework for inference of sample level disease trajectories and clusters that infer that the disease trajectories are predictive of progressions through the chronic to acute. Um, the interpretations of disease trajectory using nonlinear and condition models would give this opportunity to capture clinical, cellular, and molecular changes in myocardial infarctions. The other interesting things about pilot is that we can use it in different modalities. Right now, we focus to use pilot for multiplex images. In this way, we would use spatial molecular profiling in the same sample where histology is available. Then we can capture molecular features from histology, and then we can predict these molecular signatures from standard histologies. So for more details about this work, you can check our papers and GitHubs, and you can reach through the emails. Thanks for your attention.
Thank you. We have time for a question. Mina, I have a question, which is, are you able to resolve cascades of regulation? So maybe an early event is like activation of some transcription factor, and then do you see like known downstream targets of that rising up later on in kind of disease process?
That's a very good, interesting question. Perhaps we could do that. We just have the need ataxic data, right, to capture the activity of the transcription factors.
Yeah.
That would be interesting.
I guess, yeah, I suppose you could use some well-known targets of those transcription factors.
Yeah,
Yeah,
yeah.
okay. Uh, Thank you so much. Let's take the speaker again.